Assalamu alaikum everyone. Today our topic is the coordinate regulation of glycolysis and gluconeogenesis. So when gluconeogenesis occurs primarily in the liver, where its role is to provide glucose for export to other tissues when glycogen stores are exhausted and when dietary glucose is unavailable. That means is that when our body is in a fasting state or when our body don't have enough glucose, the gluconeogenesis is took place. So the gluconeogenesis employs most of the enzymes that act in the glycolysis, but it is not simply the reversible of glycolysis. So here one question is arise in our mind that what is the difference between the glycolysis and the gluconeogenesis? So the simple answer of it is that glycolysis breaks down glucose to produce energy, while gluconeogenesis synthesizes glucose from non-carbohydrate precursors. That means that is the process when we got pyruvate from glucose. On the other hand, gluconeogenesis is the process when we got glucose from the pyruvate. So the last step of the glycolysis is catalyzed by the enzyme pyruvate kinase, the formation of pyruvate from phosphoenol pyruvate. On the other hand, in the gluconeogenesis, the first step is the conversion of pyruvate into phosphoenol pyruvate. But there is a thing to mention that there is two enzymes work on it, because there is an intermediate named oxalic acetate is used in gluconeogenesis. Here pyruvate is converted into oxalic acetate with the help of the enzyme pyruvate carboxylase, and further the oxalic acetate is converted into phosphoenol pyruvate with the help of the enzyme phosphoenol pyruvate carboxykinase. So in the 8th step of gluconeogenesis, a very important enzyme named fructose 1,6-bisphosphotase is used for the conversion of fructose 1,6-bisphosphotase into the fructose 6-phosphate. This is the regulatory enzyme of gluconeogenesis. The last step is the conversion of 6-phosphate into the glucose with the help of the enzyme glucose 6-phosphatase. This is the first step of the glycolysis, where hexokinase enzyme was catalyzed the reaction for the conversion of glucose 6-phosphate from the glucose. So as we mentioned that the three reactions of glycolysis are very exergonic as to be essentially irreversible. These are catalyzed by the hexokinase, phosphofructokinase 1 and pyruvate kinase. On the other hand, the gluconeogenesis has a very important enzyme, as we earlier mentioned, the fructose 1,6-bisphosphotase, which is catalyzed the reaction of the fructose 1,6-bisphosphate to fructose 6-phosphate. So this has a very large negative free energy because this is ATP consuming steps. So if we summarize the bypass reaction of gluconeogenesis, the first step, the conversion of the pyruvate into phosphoenol pyruvate, which was catalyzed by two enzymes, that is pyruvate carboxylase, and it formed the oxalic acid. Then oxalic acid formed the phosphoenol pyruvate with the help of enzyme phosphoenol pyruvate carboxykinase. So the second bypass reaction of gluconeogenesis is the formation of fructose 6-phosphate from fructose 1,6-bisphosphate with the help of the enzyme fructose 1,6-bisphosphotase. The last bypass reaction in gluconeogenesis is the formation of glucose, which is our ultimate end product of gluconeogenesis, from the glucose 6-phosphate with the help of enzyme glucose 6-phosphatase. So that was for today. Thank you for watching.